Let me start thanking the NexGen 70 team for the great opportunity they gave us to come to San Francisco, to Berkeley in January and acquire the data that I will discuss today. So for the sake of time, I will only present the MRI, the imaging data, although we did acquire diffusion spectroscopy. I'm happy to get any question on the spectroscopy data, but I didn't include it in the presentation, simply because we think we got some exciting pilot data on a topic which is quite hot at the moment, which is visualizing the cortical laminar architecture and the microstructure underpinning it. And we think that the NexGen 70 really give us an opportunity to dig deeper into the microstructure of gray matter. So the gray matter tissue is composed mostly of intracellular space. And if we look at the relative contribution of the different cellular compartments to this intracellular space, the projections, so neurite, glial fibers, and axons, represent the vast majority of the volume inside an MRI voxel in the gray matter. Although a non-negligible part of this volume is occupied by the cell bodies of all these cells that we generally call soma. And these cell bodies organize in a very structured fashion along the cortical columns. And in the different areas of the cortex, they have a very different laminar architecture, the so-called laminar architecture. One very big challenge to study this laminar architecture in humans in vivo is that we need high resolution to resolve each of these layers. And so here is our ambition to see if the NexGen 70 scanner with strong gradients can help us pushing down the limits that we currently have. We typically acquire diffusion MRI at 2 millimeter on our connectome scanner in Cardiff, and try to push this down at least to 1 millimeter to start disentangling perhaps supragranular from infragranular, which is the top and the lower part of these layers. Historically, gray matter modeling in diffusion MRI focused mostly on the cell fibers because it's the most abundant compartment. And there is plenty of literature demonstrating that in the experimental regime that we usually use for single diffusion encoding MRI, a lot of the features of the fibers, such as ondulation and curvature and branching, are very much negligible. We do have a sensitivity to the sides of this fiber, axon diameter or dendritic sides, but this sensitivity is very much a delicate balance between the gradient strength. We would like to have the highest possible gradient strength and the SNR, this sigma you see in this equation. So you need both. You need the strong gradients and high SNR if you want to properly, accurately estimate the caliber of the cellular fibers. So in most of the scanners, also, for example, the connectome, when it comes to gray matter, we can assume that we lose sensitivity. We have a very limited sensitivity to this caliber, the fiber size. And so the stick model is what we go for. So all the fibers get collapsed into a cylinder with zero radius. And using this model, we can make prediction. So if this model is accurate, then at very high B value, pushing the gradient strength, we should see a very distinctive power law arising from the signal, the direction average signal normalized by the B0 should scale as 1 over square root of B. Using the Connectome 1 scanner in already 2017 and then 2020, several labs replicated a result that demonstrates that this specific power law is not measurable in gray matter in vivo in humans. It breaks there. And so this raises questions why this happened. And so we proposed, as a community, a couple of explanations. One is the presence of cell bodies, and the other is the presence of exchange effects between water inside the fibers exchanging with the extracellular space. Back in 2020, we proposed SANDY the soma and denoise density imaging to answer the hypothesis that is the soma that breaks this power law. And then later on in 2022, we introduced NexGen to account also for the exchange effects. So with the NexGen, we acquired both data for Sandy and NexGen, but in less than 2 months ago, we only had time to process and provide pilot results for the Sandy part, but we have data for the NexGen as well. The Sandy model very briefly decomposed the signal into signal coming from cell bodies and signal coming from the fibers. And we can then estimate mathematically the amount of diffusion MRI signal coming from each of these compartments, voxel-wise, and we have been comparing it, data acquired on the connectome one, with histological atlases. And we show that there is a remarkable contrast with the known cytoarchitecture of the cortex, and this has been replicated in different centers using the connectome, for example, the MGH in Boston. One of the essential requirements to use Sandy is that you need to have multi-shell acquisition, at least 5 b-values. But the important thing is that your diffusion time has to be as short as possible, around 20 milliseconds. Your small delta needs to be short, and your b-value needs to be higher than 3 milliseconds per micrometer square, which means we need the ultra-strong gradients on the next gen or the connectome. And in fact, a lot of studies have used Sandy in many diseases and in healthy development, but they used the connectome scanner. One recent work from the MGH group using the connectome 2.0 scanner demonstrated the added value of this new powerful scanner, because for the first time they were able to push the resolution down from at 2 millimeter, but to push the SNR, sorry, not the resolution. And they were able to show that Sandy intralayer contrast of cell body density was matching the staining intensity from the big brain. And same story for the neurite density was matching the myelin staining. To do this analysis, they acquired the data at 2 millimeter isotropic, and then they super resolved them at 1 millimeter, but using an AI algorithm. So they never acquired the data at 1 millimeter, simply because the connectome 2.0 doesn't have the sufficient SNR to get good data, or at least so far, at 1 millimeter. So when we went to David Thames, we wanted to push and acquire actual data at 1 millimeter cube to then produce Sandy maps and try to see if we can see this laminar architecture at the same level that was demonstrated by this very recent publication. So these are the data that we acquired. The acquisition is here, multi-shell up to 6 milliseconds per squared micrometers, with very good parameters in terms of gradient waveform, so very short diffusion time and gradient duration, very good echo time. The SNR of the raw data, it's 8, which is a workable value of SNR. It's low, but it's workable. Here you can see after denoising, we were able to boost the SNR of a factor approximately 2. However, we noticed that there is structure in the residual. This is the residual between the difference between the denoised and the real data and the raw data. And this residual suggests that there is a spatially correlated noise. So we can improve further the denoising, trying, for example, tensor MPPCA, which has been developed for this kind of noise. So this is going to be our next step. Gibbs ringing was very important to correct because we want to have a crisp contrast in the cortical layer, and you can see how well it performed. Eddy currents from FSL eddy works really well. However, we notice a marked transmit field effects, even with PTX, as you can see here indicated by the arrows. So we would like to try a different coil to try to eliminate this problem. And Sandy uses normalized direction average signal. So once you average across the direction, you boost your SNR over an additional factor 8 at IB value. So the final image looks really, really good. And although the artifacts due to the coil, we believe, are still there. So please, this can be improved. So here you see the results. These are the sandy maps, the density of cell bodies, and this is a map of the sides. The maps looks really, really good and interesting. In particular here, I recall this image was shown earlier this morning. You can appreciate the cortical characteristics of the cell body sides that seems to mirror what we know now from electron microscopy. And this is a direct comparison with the cell body density from the big brain. You can see the strike similarity, especially in the hippocampus and the deep gray matter. And to be a little bit more quantitative, and these are my last results in conclusion, you can see if we look at the profile of the intensity quantified by the Sandy and the Nissl staining, they overlap in several regions. Here you see the maxima and minima occurring at the same cortical depth. And this is another region. Considering that this is an atlas and this was me, you can see that it's quite remarkable how well the contrast match the cortical layering. And we believe this is an exciting results, and we cannot wait to work further with David and his team to push this further. Thank you very much.